Okay, so part two of conception. So we're going to talk about the process of fertilization and and then the process of cleavage, which is just a fancy term for cell division. So once where we left off, the sperm had just penetrated the egg, and it triggered the zonal reaction. And the zonal reaction was, you can see the egg is entering, the sperm is entering the egg, and the zona pellucida is starting to get dark, which is sort of our way of describing or illustrating the hardening of the zona pellucida. So the sperm, as soon as it gets through and binds to the plasma membrane, what happens is it starts to undergo some changes. So the tail degenerates or breaks off, the head enlarges, and it starts to form what we call the male pronucleus, which is basically a plasma membrane surrounding 23 chromosomes, right? The 23 chromosomes that are coming from the male. The other thing that happens with the zonal reaction that I failed to mention is that the egg is going to undergo the second meiotic division, right? So I mentioned to you earlier in the last section that the second meiotic division doesn't happen unless fertilization happens. So when the sperm enters and triggers that zonal reaction, part of the zonal reaction is that second meiotic division. And that second meiotic division is going to produce a second polar body to get rid of 23 chromosomes, and it's going to form the female pronucleus. Okay? So then we have a male pronucleus and a female pronucleus, and the word pronucleus means before the nucleus, because these two pronuclei approach each other, and then they fuse, and it forms the diploid, single-celled zygote, right? So the zygote is the result of fertilization, the result of these two pronuclei merging, and the production of the single-celled diploid offspring. Okay? Now, eventually, within 24 hours, the single-celled zygote is going to start undergoing meiotic divisions, right? And these meiotic divisions are called cleavage. So some more pretty pictures. So here, this was actually from the same book that I have all of these pretty pictures. You see the male and female pronuclei, right? So this is once it's being formed. And then over here, you see that they're approaching each other, right? And then they're going to fuse and going to form that single-celled. So they're approaching each other even more closely. They're going to fuse and they're going to form that single-celled zygote. Okay? And then within 24 hours of that single-celled zygote, you're going to see the formation of the first meiotic division and the formation of the two-cell stage embryo. So here we've got, we still have a zona pellucida. The two polar bodies are in the position of the reason why we call them polar bodies, because they're sitting at different poles. And we've got two cells that are completely identical. Okay? And the two early cells of cleavage we call blastomeres. Okay? We'll call those blastomeres. And then within every 12 to 24 hours, we see another meiotic division, which means that we go from one cell to two cells to four cells, okay? 4 cells to 8 cells. Okay? And then when we're at that 8-celled stage, again, still all of these cells are exactly the same. And this 8-celled stage is what we call the morula, and in Greek, morula means mulberry. So that's, you can see that this 8-celled stage starts to look, actually this is more than the 8-celled stage. The morula stage is beyond that. It's about the 12 to 15 cell stage. And it's about 3 days. Here we're at about 3 days after fertilization, okay, approximately. Then from this morula stage, you start to see fluid filling the center of the embryo. Okay? And then around the same time where fluid starts to fill the embryo, the cells that previously were all identical start to undergo their first major, what we call determination step, which is another kind of term for a differentiation step, meaning that they go from one single cell type to 2 different cell types. And the 2 different cell types in this stage, which we call the blastocyst stage, is the trophoblast cells and the inner cell mass cells. And this is a very important stage because the trophoblast cells are all going to become extra embryonic tissue that is not part of the embryo, including the placenta, right? So all the placental cells are coming from trophoblast. And then the inner cell mass cells are all going to be embryonic, okay, by and large. Some extra embryonic, but mostly embryonic. And it's this blastocyst stage where you have a fluid-filled core, you've got an inner mass cell layer and a trophoblast cell layer. It's this stage, this blastocyst, that's actually going to implant into the uterine lining. Okay? And the one thing that you notice from the early blastocyst to the late blastocyst is that it no longer has that zona pellucida. And when it sheds that zona pellucida, sometimes that's described as hatching, right? So the zona pellucida is analogous to the shell on this egg, and it hatches from it, right? And then it's just the blastocyst minus the zona pellucida that implants into the uterus. So over here, we just see kind of the evolution of all of these stages. So we start from the growing follicle, right, in the follicular phase early in our cycle, and it's growing and growing, and then we form the mature follicle, right, one of them wins, and then we release this oocyte. It undergoes that first meiotic division, and we release the oocyte. The follicle becomes the corpus luteum, right, and then the released oocyte is caught by the fimbriae, taken up into the fallopian tube, and then it reaches that ampulla, it gets fertilized. And then as it travels now down that oviduct or fallopian tube towards the uterus, it's now undergoing these stages of cleavage. Okay, so it goes from fertilized to the single-celled zygote stage, to the 2-cell stage, to the 4-cell stage, the 8-cell stage, the 12 to 15-cell morula stage, and then finally the blastocyst stage. And then it's this blastocyst stage, once it hatches from that zona pellucida, it's this stage that it's going to implant into the uterus. And we can see some pretty pictures of this happening. So here we've got a 2-cell stage fertilized embryo surrounded by the oviduct. Okay? And then we see it going from 2-cell to 4-cell to that morula, and then it's starting to undergo, right, the fluid is starting to fill it and starting to undergo that blastocyst formation. And over here we've got the fully mature blastocyst with the inner cell mass right here, and then the trophoblast surrounding it. And then on the bottom, what we actually see is here is that blastocyst, and it is being hatched from that zona pellucida here. Right? And it's this hatched blastocyst that's going to implant into the uterus.